Hello, so today's video is going to be on the complement cascade, and we're going to talk about the classical pathway. I'm also going to have two other videos on the alternative pathway and one on the Mannose binding lectin pathway, and I'll make a little playlist for you guys so you can have all three of them and follow along on that. So, classical pathway, what's going on? So the complement cascade, the main purpose of it is to kill bacteria or get rid of dead tissue in case of injury and things like that. And what happens is all of this pathway is used to create something called the membrane attack complex, which is going to poke a hole in your bacterial cell wall and essentially cause it to explode. So how do we get to forming this membrane attack complex from complement factors? So a bacteria invaded your endothelium, is inside your bloodstream or inside of a tissue, and it gets detected by an antibody. So we have an antigen-antibody complex forming. The antigen is your bacteria, and the antibody finds it because a B cell made an antibody that just so happened to detect that bacteria. And this is very similar to how you would treat a type 3 hypersensitivity situation. That's just a little side note for reference. Okay, so the antigen-antibody complex formed. And now this is going to go ahead and activate the C1 portion of the complement cascade. So this is a protein that just floats around in your serum. It's normally inactive, so are all the complement cascade proteins. But when you have an antigen-antibody complex forming, C1 becomes activated. And C1 has the ability to cleave other proteins in the cascade in order to activate them. So it's going to go ahead and cleave C2 and C4. Now each of these gets cleaved into two pieces. So C4 gets cleaved into C4a and C4b, and C2 gets cleaved into C2a and 2b. Alright, so the classical pathway is associated with the B versions of these proteins, the B versions. So C2b and C4b are going to continue the pathway. But that's not to say that the A components aren't important, because the A components are actually going to be vasoactive, which is going to help the pathway along, because it's going to bring all the mast cells, all the neutrophils, all the macrophages to the site of infection so they can gobble up the dead bacteria once the complement cascade has killed them. So C2A is vasoactive, and C4A is a mast cell degranulator, so it's going to also be vasoactive and dilate those blood vessels. And we'll see that when you have excess 2A, 4A, you're going to have a condition called hereditary angioedema, which is edema, so all those blood vessels are dilated, and now the fluid can leak from the blood into the tissue. And it's a big problem for some people, and we'll discuss that in just a second. But first, let's continue with the pathway. So C2b and 4b go ahead and bind to each other and form a complex called C4b2b. Pretty obvious, right? So in some textbooks, we'll actually have this as C4b2a, and I want you to note that that's just the old way of giving this protein a name. So the old way is C4b2a, but this has recently been changed to C4b2b, okay? So just take a note of that because you'll see it on Wikipedia, you'll see it in some old textbooks as 2a, but the new nomenclature is 2b. And this is also called C3 convertase. So obviously this complex is now going to act on C3 and cleave it to form C3a and C3b. Now again, this is the classical pathway, so we care about the 3b component, but that's not to say 3e is useless. So C3a is going to be a chemoattractant for those mast cells, neutrophils, and everything else. Now C3b, our head honcho over here, is very important because on top of continuing on the pathway, it's also going to be forming complex for opsonization. So when you have a bacteria and you put C3b on there, it can do something called opsonization, which is when a phagocytic cell recognizes the C3b and it's like, hey, I have a receptor for this, and so it gobbles up the bacteria. So it helps the phagocytic cells along. C3b can now bind with C3 convertase, so the 4b2b, and form something called 4b2b3b, okay? And this is called C5 convertase. And guess what that's going to act on? Well, it's going to act on C5, and it's going to cleave it to C5a and C5b. Again, we care about the B component. C5a is, again, vasoactive. So C5b is going to go ahead and bind C6, 7, 8, and 9, and together it's going to form a clump of proteins that forms the membrane attack complex. So you'll see that's also called 5B6789. Alright, so we form the membrane attack complex. We can now form a hole inside of the bacterial plasma membrane and cell wall, and all the contents of the bacteria explode, and now the remnants are gobbled up by all the macrophages and neutrophils that we've attracted using our vasoactive agents and our chemoattractive agents, which are the A components. So when this process goes awry, we get something called hereditary angioedema. And before we can understand that, we have to understand what happens when your cells undergo trauma or infection. So let's say you smack your hand on a drawer, right? You're going to go ahead and activate factor 12 and factor 12A, if you'll remember this from the coagulation cascade. Now this is important because 12A is going to activate a protein called prekallikrein, and it's going to form it into kallikrein. Kallikrein is the main honcho here, and kallikrein is going to activate kininogen into bradykinin. It's also going to activate plasminogen into plasmin. So what you'll remember about plasmin is that usually is referred to as a clot buster, so it's going to break down those fibrin clots. But on top of that, it also has vasoactive components. So plasmin activates something called C2-kinin, which is vasoactive, and it also activates C1, part of our classical pathway, which, as you see, has all these A components, which are vasoactive. So plasmin, vasoactive, bradykinin, vasoactive. So it's going to ramp up your vasoactivity, and it's going to dilate your vessels and allow for this edema to form. But lucky for us, if you don't have the mutation, most of us have something called C1 inhibitor. Now C1 inhibitor stops activation of C1. It also stops the activation of prekallikrein to kallikrein and plasminogen to plasmin. So it has these three inhibition modes which prevent the angioedema from forming in a normal person when you don't have infection or when you've sustained a minor injury that doesn't require all this vasoactivation. Some people, though, have a mutation in the C1 inhibitor, and they have a condition called hereditary angioedema. And this is an autosomal dominant condition. And what you'll see in these people is that C1 inhibitor isn't working. So what are you going to get? You're going to get a ton of C4A, a ton of C2A, because C1 just keeps going and going and going. And you're also going to get a bunch of bradykinin and a bunch of plasmin. So usually the main test done for this condition is to look at the serum and look for C4. Now what C4 will tell you if it's low is that hereditary angioedema could be a condition to consider. So you'll often see on these people really swollen hands or swollen feet, and some people will even have a very swollen face, even after minor injury or minor infection. So that's something to look out for. The other thing I want to talk about is the membrane attack complex. When you have a deficiency in membrane attack complex, you're very susceptible to bacteria that are encapsulated, because this complex is great at punching holes in capsules. So you'll get, classically on board exams, they'll test for Neisseria meningitidis, so this person will get recurrent Neisseria infections. Other encapsulated bacteria could include something like Haemophilus influenza, strep pneumoniae, so this person is going to get infection after infection, pneumonia, and lots of problems with bacterial organisms if MAC is mutated or defective in any way. Alright, so I'll see you in the next video where we'll discuss the alternative pathway. Thanks, and I hope this helped.